So hi everyone. Very hard to talk about. So very hard to talk after the number of great talks this morning, but I will try. I'm Nikolay from Lund University Biology Department, and I'll be talking about cells, these beautiful tiny objects. I want to say immediately that I've been really enjoying this meeting because I think the organizers, they found the right balance between knowledge in biology and mathematics. So I think AI is kind of mathematics for me. There are plenty of meetings where people are sort of trying to do all kinds of AI and apply these tools to biological systems, and so without a good understanding of biology, I think it's a typical reason for failure. And other way around, when people from ecology and biology, they sort of also try to naively use AI without sufficient understanding how a neural network works, and then all the stereotypes like it's a black box comes. So I think here at this meeting, the organizers really found a good balance between these two worlds, biology and mathematics / computational analysis / AI. So I've been a disclaimer. I'm not doing sort of neural networks anymore on a daily basis. But during my career, I was working with different types of data. So starting from PhD in theoretical physics, I did a lot of machine learning in biomedicine. So medical people are very open to all these crazy machine learning ideas, this is where it's blooming. Like AI in medicine is sort of super super popular. I don't know if you've seen, guys. Like the day before yesterday, DeepMind released a new algorithm, AlphaMissense, which is kind of a cousin of AlphaFold, which is claimed to predict lots of disease-causing genetic variants. So it's machine learning in general is super popular in medicine. This is where I started doing machine learning. But recently, I'm kind of switching from medicine to more cell biology and evolution and ancient DNA. So I'm not kind of doing AI very actively anymore. But hope you'll still find interesting what I'm presenting here. So this is what I'm doing. I'm looking at sequences. So those are DNA sequences. And convolutional neural networks are very useful there as well. By the way, and yes, so my peak of interest in AI was approximately between 2014 and 2020, where I also did some computer vision, and approximately here I was doing all this stuff like image classification and object detection and instance segmentation, and I was worried that it's super outdated, but luckily people are still talking Mascar CNN and UNets and GradCam. So this is what we used to do as well, like a few years ago. And I'll briefly explain so the data sets I was working with. So I was in some point was involved and interested in this Human Protein Atlas consortium at KTH in Stockholm. And it's a number, it's many thousands of images with 28 classes. And the classes, they correspond to cell compartments. So they visualize different cell compartments, and they want to kind of detect cells with special protein expressed in special compartment of a cell. And I also worked in a close collaboration with Sonia Eitz, a PI from Biomedical Center at Lund University, and her project was about lysosome cell death. So it's kind of she created a special staining method that can visualize lysosome with broken membrane, and it's sort of associated with cell death. So cell can die in multiple ways. So lysosomal cell death is one of these ways. And we were kind of particularly looking at a large, actually very large data set, 6 million images. And we were chasing those cells that were going through lysosomal cell death. This was the goal of the project. And just a few words. So those two data sets, like from Human Protein Atlas and the lysosomal cell death data set created by Sonia, they are kind of similar because they both use fluorescence microscopy. And just to say a few words about how it works. So the idea is that the cell has multiple compartments. For example, this is a lysosome. Sort of they attach a special dye to each compartment, and then this dye it emits light at a certain wavelength when a laser beam hits this compartment. Then that's a way to visualize a special part of a cell. Sort of we spent quite a lot of time on cleaning this 6 million images data set. Yeah, this is how it looks like when they visualize by different colors different parts of a cell. So it's a canonical RGB, and the green color it stands for cytoplasm, and then mitochondria and nucleus stained by blue and red color. And then you merge these channels and you get this nice images of cells. And Human Protein Atlas, it had actually 4 channels, so which was kind of a challenge at that time. I don't know, maybe now it's easier, but at that time we were doing something like like typical software will not work with 4 channels. So at that time, and the additional yellow channel, what we did, we were adding it half of this to sort of red and green channel, because sort of yellow is red plus green. So you had to do all these tricks in order to work with 4 channels. And we went through sort of pixel intensities. Yes, so it's quite easy. So when you work with cell images, it's quite easy to measure their size, their aspect ratio, but by looking at by counting cells and looking at their aspect ratio, you can actually do a lot of filtering. So, for example, by looking at how pixel intensities, how histograms of pixel intensities look like, you can sort of easily detect cells which are images that are too dark, for example, that have very little variation in pixel intensities, kind of black, they are non-informative, or there is something going on these images when this variation of pixel intensity is huge. So this is how we were detecting low quality images without actually manually going through all these 6 million images. And then we also discovered that different images, they were clustering in a special way. I don't know if you guys are into cell biology and single cell RNA-seq biology. There they use these methods like t-SNE to visualize cells where one point is one cell, but in this case one point is actually one image. And we discovered that there was some unexpected clustering of cells, and we were of images, and we were thinking what it was. Using we trained the CNN and then looked at what was the difference between those classes, and apparently some plates, so some images coming from some plates, they were had something special in their background. At least the neural network was looking at the background instead of cells, while here I think it was looking at these 2 dots. So basically this kind of analysis allowed us to figure out that there was some difference in background between different plates. Apparently, even visually you wouldn't say that there is a difference, but apparently there was. So we did this kind of diagnostic works, And here I'm sort of on purpose, I'm not concentrating on results, but rather on methods. So what you can do with this data set, how you sort of start your analysis, how you approach this huge microscopy imaging data set. And also we were lacking annotation. So, because if you have annotation, you're lucky. But we had no annotation at all, and we were chasing the cells going through lysosomal cell death without annotation. So, and then, so what you can do, you can do some unsupervised machine learning. For example, we were trying to kind of crop or detect. I mean, it's very easy to detect these very distinct objects by using simple thresholding, and we were trying to cluster them. So again, it's a t-SNE that sort of we hoped that we would see some patterns that different images would cluster, or different crops of cells would cluster differently, but it was sort of semi-successful. Sometimes it did, sometimes it didn't. But I think it's an interesting method still. If you don't have annotation, at least you can sort of try to find some heterogeneity between your cells by clustering their images or their crops. And also, as I said, we were chasing these cells that had these bright spots, which was a kind of a sign that this cell was going through lysosomal cell death. So we did test some object detection techniques, and for this purpose I was doing sort of manual annotation, so you're literally sitting and drawing bounding boxes around your cells. And then we were testing mask RCNN, which is still there, but then apparently no one is using faster RCNN. At that time we were using this RCNN as well, and we found that mask RCNN was not perfect, but was outperforming the fast RCNN a lot. So we kind of, I understand why people don't use fast RCNN. And also we concluded, so since we didn't have annotation and this approach of cropping cells was sort of semi-successful, we thought maybe we should segment the cells and crop, you know, the contour of the cells. And then we did test sort of different instance segmentation methods using actually benchmark data set from Broad Institute. And at that point, there were two popular methods popped up, StarDist and cell pose that were sort of pre-trained on cells, and they were claimed to segment cells very well, or nuclei, to be more specific, nuclei. So we were doing different types of comparison. So in order to do instant segmentation, you need to, in addition to original image, you need to have this ground truth mask. So it's kind of annotation, which was, you can't create that many masks. But for this Broad Institute dataset benchmark that we used, there were 200 images with these masks. So we were kind of looking and comparing different methods, such as thresholding, which is thresholding, which is Otsu, and then StarDist and cell pose. And also we trained our own UNet and mask RCNN. I think this is what I'm showing is mask RCNN segmentation. And sort of our conclusion was, if you sort of compare these different methods in terms of intersection or union and F1 score of classification of each pixel, and we had 4 classes of, as far as I remember, 4 classes of sort of pixels. I mean, they all were comparable, more or less. So that's why, like, training our own mask RCNN and UNet did not improve dramatically the accuracy. So that's all I wanted to say. Thanks.